Okay, so chapter 15 is adaptive specific immunity and immunization. Chapter 14, if you remember, we did the non-specific. So we're left now with the specific, which is adaptive. Adaptive means it is going to develop and it needs training to adapt to something. And it will be specific, so obviously we are talking about which cells? Which cells are responsible for specific? B and T. B and T. Otherwise, all what we talked about so far, everything, inflammation, macrophage, microphage, complement, interferon, all of that was, including the skin and everything, that was the non-specific parts. Now we're talking about specific. Specific means each one is each cell is responsible for something specific. It's not like macrophage. Macrophage, when it see anything, it will take care of it, irrespective of what it is. Is it a virus? Is it a particle? Is it a bacteria? Whatever it is, it doesn't matter. And others as well, but now we're talking about specific. Example, I am the B and T cells that are responsible for viral B hepatitis only, or influenza type 7, whatever it is. So that will be very specific. So antigen, and remember this chapter is very close to A and P, and maybe a little bit of pathophysiology, but it's mainly physiology. But there are certain parts that are very important to know, so I'm going to focus on those. But to start, what's the antigen? This is just an introduction to this chapter. The antigen is any foreign infectious agent, and when I say agent, I mean agent. I didn't say foreign bacteria or foreign virus. I'm saying agent. So agent is an umbrella that means anything. It can be virus, it can be bacteria, it can be a protein particle, it can be anything. That's why we say agent, whatever it is. Anything that is foreign to the body, infectious, meaning it's going to cause changes in the body. And because it's infectious and because it is foreign, the immune cells are going to attack that agent, whatever the agent is. What's going to happen is, if this is a microbe, let's say it can be anything, not necessarily microbe, but if we say microbe, what's going to happen to it? To prepare for the battle, to prepare for the fight, what are you going to do? The very first thing is the macrophages. The macrophages are always everywhere in the circulation, ready for anything. I usually say the macrophages, or the phages in general, these are like, you know, these police cars going around the streets all over the place all the time, and when they find something, they go to it and stop it, right? Irrespective of what it is, right? They don't think. Like, if you are breaking the light, I'm going to stop you. But if you're speeding, it's not my job. No, it's not like that, right? Even if they see a car on the side, they will stop it. They will stop and see what it is. An accident, they will go. Breaking the light, they will go. So this is non-specific, right? You go to anything that's happening, and you are the first person or the first responder to go and see and check what's happening there, right? And then when you go and find something, whatever it is, you are going to take it and eat it, right? And then you're going to show the antigen of that agent to the rest of the circulation, or of the immune system, I mean. Okay? Are we okay? Are we following? Yep. This is what's happening. Something foreign got into the body, to the blood. What's going to happen? Macrophages are going to go to it. It will find out what it is. It will eat it, which is phagocytosis, right? And after you eat it, it will show its antigen. And antigen is a foreign particle, right? To the rest of the immune system. And it's going to secrete some chemical agents or some chemical compounds to attract the rest of the immune system. We're talking about B and T, which will come, and it will be specific later on. So they are going to call the lymphocytes and show them the antigen. As usual, stop me at any time. If you have a question, if anything that's not clear, if anything doesn't make sense, stop me anytime. So when the macrophage interact with that agent, and then it's going to secrete chemicals. These chemicals are going to attract lymphocytes. When I say lymphocytes, I mean generally speaking, lymphocytes, T and B. And these, we call it the skilled troops. This is like, again, police car moving around and they found something. If they can take care of it, that's fine. But most of the time, if this is something that's very specific, like something related to drugs, for example, right? They will need to call that department of the police that's dealing with the drugs, right? Which is the skilled one, the specialized one, the specific ones, T and B. They are going to sell chemicals, and these chemicals, we will talk about it. This is just an introduction. We will call these chemicals cytokines. Cyto means cells, kines mean movers. So these are chemicals that will be released to call the specific part of the immunity, which is T and B. Okay? They will come over and take a look at the antigen, recognize it, and see who is specific for that antigen to take care of it. But the process of phagocytosis, taking that agent and showing the antigen to the T and B when they come, we call that antigen presentation. They are presenting the antigen to those specialized ones. It will come again, and again, this is just an introduction. So next is vaccine. Again, an introduction about the vaccine and other therapies that will mimic nature. What does it mean? It means if you got sick, you are going to develop your immunity the same way exactly, which is you got sick, actually sick, okay? So a microbe entered your blood, macrophages will go to it, it's going to phagocytose that agent, and it's going to present the antigen. It's called the specialized troops or the specialized cells, which are B and T, and so on. So, but the problem is those agents that entered are harmful. They are not controlled. They are actually making you sick. So they are going to make injury. They are going to injure your cells, right? So they are harmful. But when they developed the vaccine, they said, how about we make something similar to getting sick, but it's a smaller dose. It's something that causes infection, but does not cause harm. If there is a difference between those two, yes. Infection is something and pathology is something else. Infection means the microbe entered your body. We call that infection. The pathology is the actual harm that's going to happen to the cells. Are we following so far? So you're getting the good part, which is let your immune system be trained on it, but you're not exposed to the full load of pathogens that can be harmful. So this is vaccine. We talked about the first and the second line of defense, okay, which is innate, something that you're born with, most of it. But now, which was the first line, do you remember this? First line, the physical barriers. Second line is everything else, inflation, phagocytosis, complement, and all that stuff. Now this is the third line, and this is acquired. Acquired means you are going to be trained to it. You're not born with it. So you're going to adapt to something. And this is basically T and B. Now the antigen. The antigen, I talked about it. And the antigen is any foreign agent that's going to stimulate your immune system, including T and B. There are certain characteristics to call it an antigen. This part is important to remember. What are the characteristics that make you call it antigen? Why not everything is an antigen? Here is why. The antigen, number one, it has to be foreign to the immune system. And we know this part. Number two, they can be a large molecule that are non-self. It doesn't have to be a small, it can be a small molecule or large molecule. It can be a microorganism or any molecule. It doesn't have to be a microorganism. So Bacteria have antigen? Yes. Virus have antigen? Yes. Any foreign agent can have an antigen? Yes. Whatever it is. A different type of protein entered your body for some reason, it is going to cause the same thing. So is it only microorganisms? Is the antigen only microorganisms? No, it can be microorganisms, can be anything. Virus, bacteria, particle, whatever it is. And the example of that is pollen, bee venom, proteins, toxins. Are these microorganisms? Plant pollens, is that microorganism? Of course not, right? The venom, the proteins, toxins, are the toxins microorganisms? Of course not. So it can be any one of those, as long as it is foreign to the body, as long as it stimulates the immune system. These criteria that you have to fulfill, you have to look at it and see if this is an antigen or not. So you need to remember this part. We're talking about the specific immunity, and the specific immunity have two characteristics. Number one, specificity, and we said that this is the specific immunity, right? Specific immune response. So specificity means you are going to be specific against that specific agent. This one agent is my responsibility. Any other agent, not mine. So we call that specificity. This is what I'm going to respond to, that specific antigen. Number two of the criteria or the characteristics is memory. So lymphocytes, which are B and T, both of them, which are responsible for this type specific immunity, not only specific, but they are also able to create a memory. Okay? Meaning they can recall if something happened. Like if this is the first time ever that you are exposed to pollens, for example, right? You are going to do the whole process, macrophage, B and T, and everything, right? And then you will take care of it. And after that is over, it's not over for your system. It's not over for your immune system. It's not over. They are going to put aside part of your T and B to the side and tell them, remember what happened today, remember what happened this time. So if that happened again, you be ready for it. And we call that memory. Are we okay so far? Okay, the next antigen is a very special antigen that's called the MHC, or major histocompatibility complex. The other name for that is HLA, which is human leukocyte antigen. What is that? It's a set of genes that code for receptors. Okay? Major histocompatibility complex is a complex of genes. A complex of genes that code for specific receptors. We call these receptors histocompatibility complex receptors. Okay? These receptors are cell surface proteins, and these are essential for acquired immune system to know who is self and who is not self. And when you say histocompatibility, histo means tissues. So we're talking about cells, right? So it's not on agents. I didn't say agents. It's not microorganisms. We're talking about cells. Okay? So these are the ones that are responsible for self versus foreign cells. So our own cells, is that self or not? If it is not self, you're going to reject it. We hear all the time about this person taking like a kidney transplant, but unfortunately he rejected it, right? How did he reject? He rejected it because the immune system did not recognize these antigens on the cells. And what are these antigens? Or these receptors, I mean? These receptors or antigen are MHC. Okay? So what actually happened is, if you are going to receive organ transplant, you know what you're going to do? First thing is, why do they prefer a relative, especially first degree, son or brother or sister or something? They prefer that to make sure that the MHC is very close. Okay? This is one. They do blood tests to make make sure it's the same blood. But the most important step is, let me see your MHC, let me see the MHC of the donor and match them. If these are very close, you are going to receive the donation, but still, I do not guarantee. Okay? This is what happened. I get you as close as possible of your MHC. There is nothing like 100% identical. So I find like the best choice for you, which is very close MHC receptors. So and then I will give it to you. It's not 100% like yours. So I'm going to suppress your immune system temporarily until your body gets used to it. It can succeed, it cannot. I don't know. Okay? And these cells are two types, or two genes. These, I mean not cells, I'm sorry. These genes that code for receptors are two types, class 1 and class 2. And I'm going to talk about the details. So we have two different types. Simply depend on which cells are we talking about. Are in these receptors. What's MHC again? These are genes that code for receptors. Okay? Genes that code, it's a complex of genes that code for receptors on the cells, on the cell surface. So we have two types, class 1 and class 2. Class 1. Class 1 is found in any nucleated cells of the body. Any cells that have a nucleus will have class 1 MHC. And the T lymphocytes are going to interact with that class 1 to know that this is your cells. The other type is called class 2, which is found only on antigen presenting cells. This is a unique type. Are we following? So class 1, any nucleated cells, which means your body in general, any cells. Okay, liver cells, skin cell, any cells. Class 2 is only in APCs. What are the APCs? Antigen presenting cells. These are the cells that when they interact with the antigen, they are going to present that antigen to the rest of the immune system. Okay, are we following? Okay, so like macrophage, like dendritic cells, like B cells, and you need to remember these cells. So if I ask you what are the APCs, you need to tell me these cells. Macrophage, B cells, and dendritic cells. Meaning these cells, when they deal with the antigen, they are going to introduce that antigen or present it to the rest of the immune system. And we're talking specifically about presenting it to T cells. Okay? Are we following? So what are the APCs? Can you tell me the types that you remember about the APCs? Macrophage, dendritic cells, and B cells. These are presenting cells, presenting the antigen mainly to T cells. They present it to the immune system in general. Okay? Macrophage is the very first one, but not the only one. These other cells are also going to present the antigen to the rest of the immune system. So here it is. This is how it looks like, the type 1 and type 2. And where is type 1 located? Any nucleated cells. Any cell in your body that have a nucleus, which is 99% of your cells. How about class 2? Only in APCs. Okay, can you answer this question for me? Which one? C? Yes. Now how the immune system is going to react to the antigen, meaning what will happen when the white blood cells, and we say white blood cells in general because it's macrophage, B cells, T cells, it's white blood cells. When they interact with the antigen, what's going to happen? What's going to happen is the immune system It's going to react to it, like the APCs, and to present it, which is what we talked about. But we're going to talk about some details. How the immune system is developed and differentiated, what are the APCs, and what is their job. There are different types of T cells that we need to know, T helper and other types of T cells. And then what's the job of T helper cells? T helper cells are going to help. To help what? To help both T and B. Okay? T helper cells. These are T cells that are going to help the activation and development of, or activation and differentiation of, T and B, both of them, even though it's T cells, but they are going to help T and B. Okay? And this is where the name helper came from, T helper cells. So the first part is, how is our immune system develop, and how is it going to be differentiated? We're talking about the specific immune system, so we're talking about T and B. T and B are going to are developed. T and B what? What kind of cells are these? Lymphocytes. T and B are lymphocytes, right? So how we make the T and B cells? They come from the lymphocytic stem cells in the bone marrow. Both of them? Yes, both of them. We make both of them in the bone marrow. Is this part clear? And then what? They are not activated yet. So the bone marrow make inactive. Inactive. It's not active yet. T and B. The location where they are going to be activated and differentiate is different, and this is where the name came from. So B cells are going to be also activated and differentiated in the bone marrow, and the first letter of bone is B, right? So the name B and T is not coming from where you developed the cells. It's coming from where did you activate and differentiate. So where do you make both cells, T and B? In the bone marrow, both of them. Now the next step is differentiation and activation. You differentiate and activate B also in the bone marrow. How about the T? And this is where the name came from. How about the T? The T is activated by the thymus. And the first letter of thymus is T. And this is where the name came from. Okay? Then after T and B are activated, they are going to migrate to the secondary lymphoid tissues. Okay? And they are going to stay there and do their job. A good part of them. Okay? So this is important to remember. Where do we make T and B? Bone marrow. Where do we activate and differentiate B? Bone marrow. Where do we activate, or which organ is responsible for activation of T? Thymus. Okay. So here is the thymus. Or here is the bone marrow first. The bone marrow is where you make both of them. And then it's going to be activated via, in the bone marrow, which is B, and via the thymus, which is T. And then they're going to migrate to the secondary lymphoid tissues. Okay? They start, they activate, and then they migrate. Here it is. You make it in the bone marrow. Thymus activate. You go to the lymph nodes, which is the secondary lymphoid or other secondary lymphoid tissues, not the only one. Antigen presenting cells, the APCs. I just talked about it, and the APCs are the ones that react or interact with the antigen, and then they present that antigen to the T cells, to the other T cells. What are these APCs? Again, we talked about it. Macrophage, dendritic, and B cells. So what exactly are they going to do? When they interact with the antigen, they are going to modify the antigen, expose it to the surface of their class 2 receptors. Remember, what is class 2? MHC. Where do you have class 2? In the APCs. What are the APCs? Those three. You have class 2. Where is the class 1? Any nucleated cells. Anyone. So APCs, they are going to present that antigen via or bound to their class 2 receptors. Okay? So this is what's going to happen. They interact, any one of those APCs interact with the antigen, they modify the antigen, they present the antigen via their class 2 receptors to the rest of the immune system, specifically T cells. Which T cells are you talking about that are going to be activated? I'm talking about T helper cells. And T helper cells are going to help what? To help which cells? They are going to help T only?
No, B and T.
Both of them. It's going to help both of them. Are we following?
Yep.
T helper cells are going to help both B and T. So when you say they are going to help B and T, when they are activated, how they are going to be activated? These are not one of the APCs. The APCs are going to interact with the antigen first, and they are going to present that antigen to, when we say to the rest of the T cells, we're talking about T helper cells most importantly. Okay? How they are going to activate? They secrete a cytokine that's called interleukin 1, which is a chemical that's secreted by APCs. Are we following? So here you have an antigen that goes to your blood. The APCs are going to interact with that antigen. Okay? They're going to take it. They're going to modify. They are going to show it on their surface via their class 2 cells. And then they are going to secrete chemicals. And these chemicals are going to activate T helper. And T helper is going to help both T and B. Okay? T cells, the immunity that the T cells do is called T cell immunity because it is mediated by T cells. Okay? And let me tell you this. We're talking about specific immunity, right? Which cells are making the specific immunity? T and B. T will do a job and B will do another job. Both of them are doing the same thing at the end, but through different tools. What are these tools? T cells are going to interact by themselves. Okay? B are not going to do that. B are going to secrete antibody that goes toward that agent. And this is exactly like this. I always say it like immune system, I always say it's like police. So let's say it like this. We are 2 police officers and we're going to this suspect, right? One of us only will go to that suspect and try to handcuff him, right? And the other one will stay away with his gun just in case. The one that go and interact personally by himself with the suspect, handcuff him, this is the T cells. And that's why we call it cell-mediated immunity. Who is mediated? The cell itself, the T cells. Okay? How about the B? No, they are not going to interact. They are going to stay away with the gun just in case if something happens. What if the suspect have a gun, right? So they stay away and shoot. Shoot what? Shoot antibodies. Did you get the idea? So T cells are cell-mediated cells. Why? Because the cell itself is mediated in that interaction. B cells, on the other hand, does not interact as a cell. They stay away and they secrete antibodies towards the organism or the agent. Is that okay so far? The other type or the other cytokine is interleukin 2. And this interleukin 2 is produced by the T helper cells to activate other T and B. Right? To activate T. What are the other T cells? Natural killer and cytotoxic cells. Natural killers and cytotoxic cells. So T helper are going to help the activation differentiation of B and T, both of them, right? When I say you're going to help the activation of T, the other T, what are the other T? Natural killer and cytotoxic. Is that clear so far? They are going to activate natural killers and cytotoxic cells, and we will talk about those. But this is what's going to happen with interleukin 2. Okay? So what's interleukin 1 and interleukin 2? Both of these are called cytokines or cytokines, okay? Both of them. There is a difference. Who is secreting 1 and who is secreting 2? Who is secreting 1? APCs. Okay? They secrete interleukin 1, which is a cytokine. What's a cytokine or cytokine? It's a chemical signal that's produced by these cells. Who is secreting 1? APCs. These three. And then when they do that, they are going to activate T helper. When T helper come, it will secrete the other cytokine, which is another chemical that's released from the T helper cells that's called interleukin 2. Did we get the difference? Both of them are cytokines? Yes. Both of them are going to mobilize, differentiate, and activate? Yes. Yes.
So it's after like the T cell interleukin the T helper cells.
Yes. So here is an antigen coming, right? APCs will interact with it. The very first one is macrophage, but the other two are going to interact also. Okay, so far? And then when they do that, beside presenting that antigen, they're going to secrete a cytokine. Cytokine is interleukin 1. This is going to activate T helper. T helper will secrete 2. 2 is going to activate the other T, which are natural killer and cytotoxic, and it's going to secrete something else to activate the B. Are we following? So remember this. T helper are going to activate both B and T. Okay. When they activate the rest of T, that will happen through interleukin 2. Interleukin 2 is to activate the other T. But is it going to activate B? Yes, via something else. Another factor. Okay? A little bit details about how the T cells are going to maturate. They maturate via the thymus. Thymus is going to secrete hormones that's going to activate T. What's that hormone? Does anybody remember? Thymosin. 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 Thymus secrete a hormone that's called thymosin. That thymosin is the one that's going to activate T. Okay? There are different classes of T helper cells. We call it CD cells or cluster of differentiation. Like how is it going to differentiate? There are a lot of these CDs, but we need to know 4 and 8. CD4 and CD8. So what's a CD? It's abbreviation for clusters of differentiation. These are the receptors that are going to help the T cells to differentiate and maturate. Is that okay? How many types? A lot. But I care about 4 and 8. CD4 is a receptor that's located on T helper cells, while the CD8 is located on T cytotoxic cells. We talked about T helper, but we didn't talk about cytotoxic yet. Okay? Cytotoxic means toxics to the cells. So these are the T cells that are going to interact with cells and kill them, be toxic to them. And we will talk about it. And then just like B, both T and B are going to migrate to the secondary lymphoid organs as I mentioned before. So here is the details. T helper cells, what kind of CD they have? 4. Are we following? 4. T helper contain what cells? 4. And what's the cytokine that they're going to release, if you're following? Interleukin 1 or 2. T helper cells are going to secrete 2. Who is going to secrete interleukin 1? Another cytokine. APCs. I'm just making sure that you're following, okay? So this is the T helper cells. How about the cytotoxic cells that have CD8? Cytotoxic cells are going to, just from the name, cytotoxic. Toxic to the cells, meaning they are going to destroy cells. They are going to become toxic to the cells. Which cells are going to, these cells are going to be toxic to? Cancer cells. Cancer cells. Okay? How they are going to be toxic to the cancer cells? They secrete something called perforin. And from the name, perforin is coming from, what does it sound like? Perforation or hole, yes, perforation. So it's going to perforate, it's going to make a hole in the cancer cells and it's going to die. Of course, the question that comes to our mind all the time is, well, if that's the case, why people are not treated from cancer by themselves? Why the cytotoxic cells, they don't go to the cancer cells and kill all of them? The access to it. They do not have the access to all of those. They can reach to those on the surface. So if this liver have cancer, they can go to part of them. And they are going to do that, actually. They're going to secrete perforin, are going to perforate the cells, but unfortunately not all of them, a little bit from those. And they are working on that. This is very promising. They are going to train, that's what they are doing, and they're spending millions of dollars to train the cytotoxic cells in order to go to all the cells and secrete perforin, and instead of a little bit of these cells, to all of them. So they can wipe off. Hopefully, if it works, that will be a revolution in cancer. NK, natural killers. Here's what I want you to remember about this. And all what I'm talking about is important. Okay? Just try to remember it from the name so you can know the answer for any question. Okay? Get it from the name. Okay, T helper, to help, B and T. They secrete interleukin 2, which is a cytokine. Is that okay? So T helper, help, T and B. So when you hear the name, you know the answer right away. Okay? How about cytotoxic? Toxic to the cells. Cytotoxic, right? Which cells? Cancer cells by secreting perforin. Is that okay so far? Yes. Natural killers. Here's what I want you to know. Just how to know it from the name. Natural killers. They kill the natural cells when or if they are infected by virus, or also if they have cancer, and the third one, if these are foreign cells. So natural cells means a cell, membrane, nucleus, organelles. We call that the natural cells, right? And these are going to kill the natural cells. Which natural cells? Natural cells that are infected with virus, natural cells that have cancer, natural cells that are foreign to the body. Is that okay? So natural killers kill the natural cells. When? 3. When? When they are going to kill the natural cells? If they have cancer, virus, if they are foreign to the body. In any one of these scenarios, these are natural cells, just a cell. With everything that we call it a cell, it will kill it. If any one of those 3 happen. And then memory cells, and the memory cells is for the secondary immune response. What does that mean? The primary immune response is when you are exposed for the first time. Okay? This is your first time ever to be exposed to whatever it is, to measles, for example, whatever. Okay? Chickenpox, anything. This is your first time. For the first time, all that scenario that I talked about will happen, right? And then you are going to put memory cells on the side. So if that virus come again, now we call it the secondary response, you respond faster because you have memory cells. All right? You are trained. You have the memory cells waiting for you. These memory cells, they remember exactly that fight. And once they see that agent again, oh, they remember right away. Yeah, I remember. We dealt with this type before, and I know exactly what we're going to do. That's why it will be faster. Secondary response. So memory cells are for secondary response.
So do actually the memory T cells and what's the difference between the memory T and the memory B?
Both of them are memory cells. Both of them remember the fight. But T cells will remember how they are interacting individually because they are T cells. And B cells remember what kind of antibody did you secrete. So both of them are helping each other, and both of them will stay on the side. Each one will tell you his part of the fight and what should we do. Okay? And that explains why the secondary response is a lot faster, because they will tell you right away. Once they see the agent, they will tell you, oh, I remember, this is what happened. So do this right away. You don't have to think about it from scratch, right? As I mentioned before, T helper cells are going to secrete interleukin 2, and interleukin 2 is going to activate what? natural killers and cytotoxic cells. This is interleukin 2. How they are going to activate B, not interleukin 2, not the same one. Interleukin 2 is to activate the other types of T, but they are going to secrete another agent. Whatever it's called, B lymphocyte activator, whatever the agent is, but it's a different cytokine. What cytokine in general means, or cytokine? A chemical that's released from cells to interact with other cells. This is basically what it is. Or to interact and mobilize. Let me say it this way. Okay? To interact and mobilize. B cells are responsible for antibody mediated. Do you remember when I told you the difference between T and B? T is responsible for what type of immunity? Cell mediated or antibody mediated? T. Cell mediated. How about the B? Antibody mediated, right? They are going to respond differently. Antibody mediated is also called humoral immunity, and you need to remember this, humoral. Humoral is the same as antibody mediated, and I put it here under the that part to remember. And humoral means in the circulation. Okay, so the antibody is circulating, is moving around in the blood. So this is where the name came from. Okay, B cells, do they have memory? Yes. Memory T and memory B. T and B are involved in the fight. When the fight is over, both T and B are going to leave memory cells to remember that fight, and we'll tell you about it next time, which we call it the second response. So who is responsible for the second response? Memory T and memory B. Each one of those will tell you his part of the fight. Is that okay so far? What are the B cells responsible for? What kind of immunity? Cell mediated or antibody mediated? B, antibody mediated. What's the other name for antibody mediated? Humoral. How about the T cells? Responsible for? Cell mediated. Okay. Can you answer this? E, activate both of them. Yes. Do they secrete antibodies? Of course not. T cells does not even secrete antibodies at all. It's B. Okay. Directly destroy the target cells? No. This is cytotoxic, not helpers. Activate B? Yes. But it say only. No. Not only. T only? No. Both of them. So E is the right answer. Just one more thing to remember about B. B is responsible for what type of immunity? Humoral or antibody mediated. Okay? Here is one piece of information that's important to remember. Before secreting the antibodies, they change to plasma cells first. Remember this. So B cells secrete antibodies, yes, after they maturate to plasma cells. Is that okay? B cells secrete antibodies. Yes, they are antibody mediated or humoral. Okay. Do they secrete antibodies? Yes, when they change to plasma cells, which is the mature B cells. Clear? Is that clear? Okay. This is a diagram showing you what's happening here. The B cells, they are activated, interact with the antigen, and they start to secrete antibody, which we call it the antibody mediated or humoral, versus the T, which are going to interact in another way, directly interacting with the cells. Here is the B cell proliferation. Interact with the antigen, and then remember that who is responsible for proliferation of B? Like we're saying, B will proliferate, become plasma cells, and secrete antibodies. Are they then doing that automatically, or there is something that comes to them to be activated? What is it? Which cytokine or which cytokine? Interleukin 2, right? So they are not going to do that automatically. B cells, yes, I know that B cells will become maturated, and I know that they become plasma cells and secrete antibodies. That does not happen automatically. The T helper cells have to secrete the cytokine in order to do that. Okay, can you answer this for me? which one is it? Macrophages. Macrophages is one of what? Of the APCs? So which MHC? One is in every nucleated cell.
Oh,
It's 2. Remember this? Where is MHC1? All nucleated cells. Where is 2? APCs. What are the APCs? Macrophage, dendritic cells, and B cells. Are you following? I'm trying to get you. When you leave this, this lecture, you should remember the important parts. And this is what I'm trying to do. So to make it easier for you to study. Okay? Okay, can you answer this one? Stimulation of B cells to divide and maturate and basically to become plasma cells. Who is doing that? T helper cells. Yes. Okay? Another one. C, which is cytotoxic, right? Okay, yeah. Cytotoxic, toxic to the cells. They kill the cells if they have cancers. They secrete periphery, right? Okay, now the B cells. The B cells are responsible for which type of immunity again? B. Antibody mediated or humoral. I'm talking about B, right? So they are going to secrete antibodies, right? When they are activated. Okay. The other name for antibodies is immunoglobulins. So this is synonymous to antibody, immunoglobulins. If you think about it, what's the antibody? It's an immunoglobulin. What do you mean? It's a globulin, which is a plasma protein that is working with the immune system, or is part of the immune system. This is where the name came from. So the antibody is immunoglobulin. This is the nature. What immunoglobulin means? It's a globulin, meaning it's a plasma protein, but it's involved in the immune system. Okay? It's basically a glycoprotein that's composed of 4 polypeptide chains. So if you look at these chains, see those 2 small ones, 2 light, and the other 2 are heavy, 2 light and 2 heavy. So 4 altogether making the immunoglobulin, and this is the chemical structure. Okay, it's glycoprotein. So it has 4 polypeptide chains that are connected by disulfide bonds. Okay, and they have any antibody or any immunoglobulin will have 2 fragments. 2 fragments. The antigen binding fragment and the crystallizable fragment. You have 2 fragments or 2 arms. We call it Fabs. The antigen binding fragment, we call it Fab. Okay? Fab means fragment that's antigen binding. Okay? So antigen binding fragment, what is this? It's one of the arms which involve part of the light and heavy chain that I showed you, and this is where the antigen and antibody join together, this fragment. So this is the part that deal with the antigen. The other one, the crystallizable one, this is a constant region that is located in the cells, and it's just constant part that does not change. So I always have 2 fragments. One of them is constant to the side, and the other one is going to help me to interact with the antigen. Okay? So remember, the immunoglobulin is the same as antibody. Antibody is immunoglobulin. Okay? All right. It consists of 4 different parts. 2 light and 2 heavy. Right? And it has 2 fragments. One of them is crystallizable, which is a constant part. It does not change. And this is the one that's going to bind to different cells. And the other one is the one that's going to deal with the antigen. And we call that antigen binding fragment, or FABs, versus the crystallizable fragment, which is FC. F is for fragment, C is for crystallizable. This is a constant part to deal with cells, not antigen. Is that clear? This is how the antibody or the immunoglobulin is going to interact with the cells with their antigen or whatever the agent is. So here is the structure again, and this is the Fab. Look at the Fab and look at the FC. This is a constant part. To this part, interact with cells. This is the Fab. This interact with antibodies. You see the ends? These ends and these ends. These are going to interact with antigen. And this part interact with cells. Okay? Fab versus this versus FC. So when they interact with an antigen, if the antigen is of a good fit, this is good, it's going to deal with it. If not, like look at this fit, this is perfect, right? Look how it is fitting here. This is my antigen. This is a partial fitting or poor fitting. It's not going to work with that one. This is how they recognize the antigen. Okay, and this is obviously the Fab one. Okay, can you answer this concept or question for me? Which is answer D. Do you agree? Yes. Antibodies are produced by B cells after they differentiate to become plasma cells. We talked about the characteristics of the antigen before. And immunogen. Immunogen is anything that generates or results in the formation of immune response. And we talked about the antigen. An antigen is a foreign body that stimulates our immune system, specific lymphocytes. So what if we say antigenicity? Antigenicity. The antigenicity is the property of the antigen that's causing the interaction. Antigenicity. So you say antigenicity of this organ, meaning how is it going to become antigenic? How is it working as an antigen? If this antigenicity depend on the size, the shape, the accessibility. If it is big, it will make a bigger response. If it is small, it's going to make a small response. So antigens are different, and each antigen will have its own antigenicity. What does that mean? This antigen have its antigenicity. Maybe its antigenicity is weak antigenicity. Okay? Is it antigen? It is an antigen. But how is it going to stimulate the body and how the body is going to react? Is it a huge, big thing or a small thing? So this is the antigenicity. Is it very antigenic or not? Okay? So being an antigen, it's going to stimulate the immune response anyway. Are we following? It's an antigen and it's going to stimulate the immune response anyway. But how much? Okay? How bad it is? Antigenicity. The antigens, they have their antigenic determinants. We call it the epitopes. And these are small molecules that the lymphocytes will know. Okay? Antigenic determinant, which is the epitopes. Now there is something called hapten. Hapten came from half antigen and you mix them together. Hap means half. Okay? 10 is the last part of antigen. Okay? Hapten is half antigen. So it's not a full antigen. It is not a full antigen. What do you mean by half antigen? Is it antigen or not? It's half antigen. What does it mean? It means if this is a full antigen, it's going to, by itself, it's going to go to the body and it's going to do all what we talked about. Go to the body, macrophage, APCs, interleukin 1, interleukin 2, and a whole process will happen. Hapten does not do that. Hapten will get into the body, nothing happened yet. It has to go and settle down and go interact with something before it's elicit or it stimulate the immune response. And the best example of the hapten is penicillin. Penicillin is a hapten. We know some people have allergy toward penicillin, right? When you go to the doctor, they always tell you to ask you, right? of the nurses. They ask you, do you have allergy toward any medication? They usually ask specifically about penicillin, right? Do you have allergy toward penicillin? What if you have allergy toward penicillin? What's going to happen? When you take the penicillin as a medication, it goes to your blood. Nothing happened yet. It's not a full antigen. Are we following? And then after some time, it will go in the circulation and go interact with some cells. After it interacts, it will start the immune response. So by itself, it doesn't stimulate the immune response. It has to interact with its other half. Did you get the idea? A full antigen is capable of, once introduced into the body, the whole immune response will happen. Hapten is different. You have to get the other half. So you have to go interact with certain cells. And now the immune system will recognize you. So the immune system will not recognize that hapten until it interacts with something, and now we can see that this is something that's foreign and we have to deal with it, which is not supposed to be happening, but if you have allergy, that's what's going to happen. Once it interacts with something, the allergic response or the immune response is going to be stimulated, and it's going to start. There are functional categories of the antigen. There is something called alloantigen, and there is something called superantigen. Allo means same. Alloantigens, like the antigens of humans, we call those alloantigens. These are the markers of some members of the same species. Like humans have their own alloantigen, monkeys have their own alloantigen, okay, plants have their own, and so on. Every species have its own alloantigen. But as humans, the antigen in humans, we call it an alloantigen. And it's not in any other species. Like your antigen will not be in other species. Did we get the idea? This is what we call the alloantigen. Like the alloantigen of red blood cells, for example, the one that's responsible for, like MHC, major histocompatibility antigen. This is an alloantigen. Why? Because it's in humans. It's not in any other cells, any species. Other species will have their own alloantigen. So this is the alloantigen for these animals. This is alloantigens for humans, okay, and so on. This is alloantigen. And this is the one that's responsible for blood transfusion or organ grafting. So of course, when you take something, if you take blood, you take it from human, right? So they will have the same alloantigen, or not the same. I mean, they will have an antigen, and we call it alloantigen. What's a superantigen? Superantigen is a strong stimulator to T cells. So this superantigen, why did you call it superantigen? Because it's going to cause massive response, super response, okay? Exaggerated response. And that's why we call it the superantigen. Like toxic shock toxin. This toxin that's released from certain types of bacteria is superantigen. Why? Because if it is an antigen, it's going to, if this is a regular antigen, it's going to stimulate a normal response. Like macrophage will come and you will have a fight and all that stuff, but it's not in a huge scale. But this one is fatal. Like once it goes into the body, it's going to make a huge fight, not that regular fight. Toxic shock toxin is one of them, superantigen. Allergen means an antigen that can evoke allergic responses. So there are some antigens that does not evoke the allergic response. Okay? And there is something that can be allergen to you but not to me. Like if I go to the backyard and I'm exposed to pollens, nothing is going to happen to me. Pollen is an antigen, yes, but not allergen to me. It's allergen to you. It can generate allergy in your body, not mine. Get the idea? Did you guys get the idea? Allergen. 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 Generating allergy. It can generate allergy. Now, regarding the antigen-antibody interaction. When the antigen-antibody interact with each other, what's going to happen? 4 things are going to happen that we need to know in details. Okay? Or at least we need to know the result of each one of those. So here is an antigen came, and the whole process happened, right? Antigen, macrophage, cytokine, interleukin 1, T helper cells, 2, until you go to the B and the antibody is produced, and then it will interact with antigen. Do you know the story? You know the whole story that happened from macrophage until the B cells become plasma cells and secrete antibody. Is that scenario clear? Is that scenario clear? From macrophage or APCs until you get to the antibody secreted and interact with antigen. This part is clear, right? Okay, so what happened after that? When the antigen, the antibody binds or interact with the antigen, what's going to happen? 4 responses are going to happen now. Opsonization, neutralization, agglutination, and MAC. MAC is something called membrane attack complex. So what's opsonization? Opsonization, when you opsonize something, means you put a coat on it. This coat will make it recognizable by the phagocytes. So phagocytosis will happen. Did you understand this? Here is an antigen, here is the antibody come. The antibody is interacting with antigen, it will make a coat around it. When you coat it, the phagocytes will come and eat it. Clear? So this is a result of opsonization. You coat the whole microorganism. Okay? The number 2, and you call this antibodies opsonins. These antibodies that make opsonization, we call it opsonins. Are we okay so far? Antibodies are making opsonization. We call it opsonin. The second is neutralization. What's neutralization? It means you're neutralizing the active site of the microbial enzymes. You neutralize it. So it does not attack anymore. Of course, that's all for our protection, right? When you coat something so you can eat it, you're getting rid of it, and that's good. Neutralize it means make the enzymes neutral so it does not attack us. This is 2. 3 is agglutination. And agglutination is antibodies cross-linking the cells or particles into a large aggregate. So agglutination means aggregation. Agglutination means aggregation. What does that mean? It means like this. Look, this is an antibody, okay? And this antibody is going to, this part will deal with one cell, okay? And this, another cell, another cell, another cell. So it's making aggregation, a group of cells that are cross-linked versus the antibodies. So now we have a big aggregation. So you can deal with it faster, right? Like if you like in the street and you see somebody and you start to fight with him and a group of people come, now this is a big aggregation and everybody can see it, right? And these are cross-linked. Then you're going to do complement fixation. You are going to activate the complement, and we talked about the complement before in chapter 14. You're going to activate the classic one. So the cells are going to rupture because of the complement. Last one is precipitation, which is aggregation of the particulate antigen. That antigen is going to aggregate and then it's going to precipitate. It's going to settle down.
So when do agglutination result in the clumping between the dose and the red blood cells?
What is it again?
So agglutination, I've always knew it as clumping between the bacteria cells and the red blood cells. But when does that happen?
After the antibody is produced, like this. Here is an antigen, okay? Here is antibody coming. And I'll act with it, okay? And cells are going to come over and make a big gathering here. The antibody is going to cross-link. You get the idea? This is antigen, this is antibody. Antigen antibody come together like this. Is it okay so far? And each one of those will make a link with a cell. So cells are going to attach to this antigen antibody complex. So when does it happen? After the antibody is secreted. So the antibody is the one that's doing this agglutination. Okay? So here is opsonization. When you opsonize something, meaning you make a coat around it, and the macrophages are going to eat it, or the phages in general are going to eat it. Neutralization, the next part, the antibody block and neutralize the enzymes. Agglutination, as you see here, can you see this? The antibodies are linking these cells together to make a big gathering or to make an aggregation. Complement fixation, and then precipitation. When they come together, they're going to precipitate down. Okay? Classes of immunoglobulins. We have different types, 5 different types of immunoglobulin. This part is very important, and I will tell you a couple of things about each one of those that you have to memorize and remember very well. Okay? Very well. What are the different types of immunoglobulin, which is antibodies, meaning? There is GAMED, gamete. Okay? GAMED. The first one is G. What do you need to know about G? This is how I remember it. Okay? G, great. The first letter of great. This is not how it goes. I'm just trying to help you remember. G is for great. So this is the most prevalent. This is the biggest amount of these immunoglobulins. And this is responsible for the secondary immune response. The third one is, this is the one that goes from the mom to the fetus via the placenta. And it also activates complement. You have 4 things that you have to remember here. Okay? You have to memorize this. What do you need to know about G? Number 1, great amount. So this is the most prevalent. We have a lot of those. This is the major part. Number 2, this is the one that crossed the placenta from the mom to the fetus. Number 3, it's responsible for the secondary immune response. Number 4, this is the one that activates, or it's one of the activators of the complement. Did you get the 4 points that you need to remember about G? A is surface and secretion. Okay, I remember it ASS. I will not say it, but this is how I remember it. ASS. Okay? A is responsible for S and S. A, surface secretion, meaning the antibodies on the surface are A. The antibodies in the secretions are A. Example, what kind of, did I say antigen? Antibody, the type of antibody. So here is an example. Can you tell me what kind of antibody in the breast milk? No, you're talking about pregnancy, but isn't breast milk is a secretion? It's A. What kind of antibody in the tears? A. It's a secretion, right? What kind of antibodies do you have inside your mucus, on your mucous membrane? A. Isn't that surface? Yes. Isn't the breast milk a secretion? It is. Sweat. What kind? A. Isn't that a secretion? ASS. So just you put it together. Okay? But this is how it helped me remember it. Okay? M, the way I remember it is momentum or moment. In a moment. This is the one that appears in a moment. Meaning it's the first one to appear. Okay? It's the first one to be secreted. The first class to be secreted. The first one that we make and the first one to be secreted. It also activates complement. But this is the most important. This is how I remember it. M for moment. It's secreted in a moment. So it's the first one to be secreted momentarily. Did you get it? This is how I remember it, and I think you can remember it as well. The next one is E. Immunoglobulin E for allergy. E for allergy and parasite. Immunoglobulin E for allergy and parasites. Okay? D is not the most important. This is just a receptor on B cells, just a receptor. Okay? So there are 4 things to remember about G. Okay? 4 about G. What is it? 1. Great. So this is the most prevalent one. This is 1. Number 2, secondary response. What's the secondary response? Do you remember the first and primary and secondary response? This one is for secondary response. The M is for primary response. Are we following? Do you remember the primary response? You respond for the first time, you secrete M. Next time if the same happen, you secrete G. Okay? So secondary response, this is number 2. Number 3, placenta. This is the one that go from the mom to the fetus via the placenta. It also activate complement. So just remember that both of them activate complement. Both G and M activate complement. It's not something unique. Both of them activate complement. Just put it to the side. Okay? But the other ones you have to remember. How about A? Surface secretion. Surface and secretion. M? Yes, M, moment. M, moment. The first one to be secreted. And primary response. First one. So it is a primary response. E, allergy and parasites. D is just a receptor on the B cells. Okay? Can you match those? Which one is secreted most when exposed and when exposed the first time? M. Which one is responsible for secondary? G. Which one is responsible for allergy? E. Parasite. E. Allergy and parasite. E. Allergy and parasite. Which one is secreted in the milk? A. ASS. A for secretion and surfaces. Which one cross the placenta? G. Which one activate complement? G and M. Right? Did you get this? This is very important slide. Okay? Antibodies are always located in the blood. It's always there. You secrete it for some reason, and then it builds up and stay in the blood for some time. This is the antibodies. And this is where the term antiserum came from. Antiserum is the serum containing specific antibodies. Meaning, and this is the actual use. Here is a horse, a monkey, an animal, whatever the animal is. Usually it's the horse, okay? If you expose the horse to any agent, not necessarily you make it sick. I mean, you expose it to a little bit of an agent, right? It's going to do the process, right? Similar to ours, okay? When that happens, he will end up making or building up some antibodies in his serum, right? Okay? How about if we take that serum containing the antibodies that already made and just give it to you? We call that antiserum. Sometimes it's needed, like if you are very sick and your body is weak. I can't expose you. You're already sick, right? And you are trying to fight, and I'm giving you antibodies and everything, but you're not fighting that well. I can give you some antiserum, so it's contained the antibodies that already made. You don't have to fight to make it. The horse already did that for you. Okay, so we make it in an animal or something, and we collect that serum that contains the antibody that's ready to use. Normally, the way you do it is that you have to go through the whole entire fight, right? Starting from the macrophage until you do the antibodies. You have to go through the fight. How about if I give it to you that's ready to use? Sometimes it's useful if you are fighting and you're very sick So why don't I give you the anti-serum? And we will have some details to talk about later. Can you answer this question? The Fab, right? Yes. Primary and secondary response. Which one is faster? Secondary is faster. You already know. How did you know? How can you remember? Because you produced T and B memory, both memories. So because you have the memory, it will be faster and more effective. Okay? So primary response is the very first time that you are exposed to that antigen. Okay? So it will take some time to develop. It will contain a latent period until you end up producing the antibody. And which is the first antibody that is going to appear in a primary response? M. And then G will show up later on. And then after that you will have the memory cells, which will help you in the secondary response. So comparing primary to secondary. Primary is weaker, isn't it? It stays for a short period of time. I'm talking about the M. Okay, I'm not talking about the secondary response after you make the memory. I'm talking about in the very, very beginning. Okay, so it is weaker than the second response. Short, it stays for shorter time than the second response. Slower than the second response. It's IgM. Is that clear? So the primary response, which anti-M immunoglobulin? Primary response, G or M? M. Is it faster or slower? Slower. Is it weaker or stronger? Weaker. Does it stay for longer time or shorter time? Shorter time. Great. And the opposite is when you come to the second response. The second response is a response of the memory lymphocytes. When you say memory lymphocytes, I mean both of them, memory B and memory T. Okay? So what will be your immunoglobulin here for the second response? G. Is it stronger or weaker than the primary? Stronger. Does it stay for longer time or shorter time? Longer time. Is it faster, quick, or slow? Quick. Which antibody is helping you with the second response again? G. Monoclonal antibodies, these are some antibodies that have single specificity. We call it monoclonal. There are others that can work against more than one. But if it is single specificity, I can work on antigen, this antigen only. That's it. Some of them will be cell specific, but I can work on this and another one. But if it's working on one only, I call it the monoclonal ones. This is a list of names. All of them would end with MAB or monoclonal antibodies. And this is useful for diagnosis of different issues, not necessarily cancers, issues. Like Herceptin for breast cancer, and this is where the name came from, the name of the medication here, which is the chemotherapy. The trastuzumab, and all of them will end with MAB. B cells, they will have that MAB, monoclonal antibody, that if you see it, you will know that this is the issue. So if you see one of those, you know what's the issue, okay, from that MAB. What's the MAB? Monoclonal antibodies. Are we following? What's monoclonal means? It's monoclonal antibody is antibody that's mono. Mono means single specificity. So if you see it, like Xolair, this is asthma, okay? and so on. So each one of those will show a specific map, MAB. If you see it, you can diagnose. And this is a list of medications that work on that map and work against that disease. I'm not going to ask you to remember those. Take a look at it and just understand the concept. Okay? Too much to remember. This is not pharmacology. So just remember what's MAB and take a look at it to just have an idea about it. But just remember what's MAB. Okay? Monoclonal antibody, single specificity. And it's helpful for diagnosis because it's single specific. Meaning, if that's not single specific, I mean, it can be 2-3 things. How would I know, right? But if it is one only thing, when I see it, I know the condition. Did you get the idea? More than that, it will be too much details. Now, the different categories of immunity acquisition, or how are you going to acquire the immunity? This part is also important, so pay attention. The immunity can be active and can be passive, can be natural and can be artificial. And here is what we mean. Active immunity means you are going to be active. You are going to make the effort. And you're going to go, meaning what do you mean by effort? I mean, you're going to start with the macrophage and interleukin 1 and T helper cells and interleukin 2 and the rest of the whole fight scenario. Isn't that active? Right? It's not, I don't have something that's ready to work, like anti-serum. That would be passive, right? You didn't do anything. I just gave you antibodies. What did you do? This is passive, isn't it? So active means you're going to be active. And you're going to do the whole process starting from the macrophage until you produce the antibodies and making the memory cells. Is that clear? So you're active, meaning I'm doing all that effort. Active. Clear? Passive, on the other hand, you're not going to do all of this. I will give you the antibody that's ready to use. Okay? Exactly like the difference between passive is like you call Pizza Hut and order for pizza delivery. Did you do anything? Or just going to eat it? Right? Did you do any effort? Nothing. Just make a call. They did everything for you. So this will be passive. On the other hand, what if you get the flour and the eggs and you mix them together and do everything from scratch? Isn't that active? You're making everything, all the details, right? So this is how it goes. So this is the first part. So when I give you scenarios, you will need to tell me. Question number 1, is it active or passive? Did you do the effort or not? Or I give you something ready to use? This is 1. Number 2, did that happen naturally or artificially? Meaning, natural means something that happened through normal life experiences, something that happened by itself. You didn't mean to do it. You did not induce it. Okay? Or is it something artificial? Example of artificial is vaccine. Is that natural? Does that happen to everybody? Or you choose to do it and you induce it, right? You decide to go to the pharmacy or to the doctor and ask them to give you the vaccine. Is that something natural that happened to everybody? No. You choose to induce it. You choose to acquire it. You choose to artificially take it. Did you get the idea? How about if you get sick? Is that natural or artificial? Natural. Everybody can get sick, right? So this is the idea, and you have 4 different scenarios to think about. So natural is just happen through life experience. It just happened. You got sick. What are you going to do? Active, your immune response do from A to Z. Passive is something that you get ready-made. Here is an example. Breast milk. Breast milk contain antibodies. Did the baby do anything? 0. Isn't that passive? Yes. What if you got sick? It's active, right? Is it? Yes, it's active and natural at the same time, both of them. So you have to tell me both cases. So I'll give you some scenarios and you tell me. Okay? Okay. You got sick. Natural, active. Did you get the 2 components? You have to give me 2 answers. Natural and active. This is scenario number 1. Scenario number 2, vaccine. Artificial active, yes. Breast milk. Natural passive. How about if you are in a hospital and I give you anti-serum, IV? Artificial passive, right? This is not something that's happening naturally. You go to the hospital and they decide to give you something to help you, right? So did you get the scenarios? Yeah. So this is artificial, something that's induced. It's not something that's naturally happening. Active and passive. Okay. Can you answer this question for me? Which one is natural, passive? The first one, natural and passive. Isn't it? Yeah. Is that easy, the 4 scenarios? Okay. Now vaccine. Is vaccine active or passive? Vaccine. Active. Is that artificial or natural? So it's artificial, active, right? Do you know what happened? Did you take a vaccine before? You get the vaccine and you have issues, right? You have redness and you get a little bit sick and you feel like some fever, and right? So why? Because you are doing the whole scenario, but on a smaller dose, on a smaller level. Okay. So where do they get the vaccine from? They can get it from whole cell or virus that's killed or inactive. Meaning, flu, for example, if you think about it, what's flu? Flu is, they get the virus, the flu virus, and they kill it. So how is it killed and still doing the job? Because the antigen didn't go anywhere, right? It's still, the antigen is still there. It's killed, but the antigen is still there, right? So if I give you that vaccine, you are going to react because you saw the antigen, but it's not, it's dead. So it's not alive, so it's not going to make you sick. Did you get the idea?
So, I forgot the MHC, the major
Compatibility antigen.
Yes,
Yeah.
where is it located? On the antigen cells or on the...
This is a gene that co-receptors on type 1 in nucleated cells, type 2 is APC. But we're talking about receptors here, okay? We're not talking about antigen, this is receptors, okay? Antigen, I can get a virus, kill it, make it inactive, and then I give it to you under the skin, for example. Does it still have the antigen? Yes. So is it infectious? Yes, it's causing infection. But is it going to cause disease? No. Did you get the difference between infectious and pathogenic? These are 2 different terms. Are we following? Infectious means it can stimulate the immune response. That's it. We call it infectious. Causing the harm, causing the damage, this is the pathogenicity, or this is the pathogenic part of it. Okay? The pathology that's going to happen. So I can get the whole cell and kill it, or get a virus and inactivate it. This is one. Or I can get live attenuated. I don't really kill it. I just make it weaker. Okay? I expose it to certain chemicals to be weak enough. Okay? What determines this or that? It depends on the antigenicity or the pathogenicity. Like in influenza, they know that this is a strong virus, so I'm not going to make it weaker and give it to you. If I do that, it will activate itself and infect you. So I'm not going to do that. For flu, because I know this is a bad virus, this is a strong virus. If I make it weaker only, it will become stronger again. I'm not going to do that. I'm going to kill it. Okay? Other viruses are not that strong. So it's enough, or bacteria, whatever it is, I can just make it weaker. And I know for sure that this is, if I make it weaker, that's not going to cause harm for you. Depending on, you can call it the strength or the power or whatever you want to call it. Okay? It can be also a cellular vaccine, like not even a cell at all. I can extract the antigenic molecules from the bacteria or virus. Example, if you're getting vaccine against meningococcus, we know that there are vaccines for meningitis, right? Meningitis, the meningococcus, which is responsible for meningitis, is a strong bacteria, okay? So I cannot kill it. I'm not going to kill it, and I'm not going to make it weak. No, none of those. I'm just going to take a little part of the capsule, okay? Just a little part of the capsule. We call that acellular vaccine, and I gave it to you. That's not even a bacteria, so there is zero chance that's going to cause a disease. It's not even a bacteria, and not even a dead bacteria. Not even that. No, it's just a little piece of the bacteria. Is that clear? Same for the pneumococcus. The last one is completely genetically engineered. We call it the recombinant vaccine. Okay? I know how the antigen looks like. I'm not getting the virus. I'm not getting the bacteria. I'm not going to kill it. I'm not going to make it weak. I'm not even part of it. None of those. I know exactly how the antigen looks like. I know the components of the antigen, and I'm going to generate that in the lab. Yes?
So when did you get a vaccine including, like for you to have the first primary response, like at a less elevated, like sickness, I guess, like you're not gonna be, like, you'll be getting it, but not on like a big scale. Is that right?
Kind of. Yes. It's going to start the process,
Okay.
but without the harmful effect of that microbe.
Okay.
Because if I give you that bacteria itself, yes, it's going to do the primary response, and then it's going to attack your cells. So you're avoiding the bad part.
Okay. And then what is the can stimulate the immune response? Like what is that for?
If it can stimulate the immune response, we call that infection. Infection is not necessarily causing pathology. Infection is you stimulate the immune response to work. That's it. Did you cause any harm? I don't know. If you cause a harm, this will be a pathology, something else. So what happened to the vaccine is it's an infection that's going to cause you to develop or to activate your immune response from A to Z, right? But there is no pathology. Even though there are some exceptions, but this is not our issue. Like sometimes, rarely, especially with the live attenuated virus or cells, if it is attenuated and your immune system is kind of weak, they can activate themselves and cause actual infection. Just know that for your information, but this is very rare. Here is a checklist, and this checklist is important to know. How to call this an effective vaccine. Like, for your information, they came up with a lot of vaccines. Some of them was accepted and others was rejected by the FDA, right? They have a checklist. So you're coming up with a vaccine. I have a vaccine. I would like to register that vaccine, and I want it to be used for public. They will tell you, okay, give me that vaccine and let's talk about it. Is it easy to administrate? If it is easy, like yes, I can put it under the skin. Okay, check number one. That's good. How about the adverse effects? Is it going to cause a lot of issues to the patient? They will say, no, it will just cause like some redness and maybe a little bit of anorexia or something. They will say, okay, this one is fine. Okay, is it going to cause a big issue? Yes, no, reject it. Okay, did you get the idea? These are the checklists that they actually use. If you're coming up, some vaccines are rejected. If you're wondering why some vaccines are rejected, this is why. They go through it one by one. So adverse effect is number two. It's never been zero, just for your information. There is no zero side effects. It's not zero. Even the redness is one of the adverse effects, right? But it's a very low level. And it should protect against both natural, wild, against natural wild form of the pathogen. So when you give it to me, you need to make sure that if I am exposed to the actual natural wild form, You're not giving me a theory, right? You're giving me, like, here is a vaccine that if you take it, you will be immune against flu. Which flu? The one that we actually know, or you're talking about something that you know, and it's not even prevalent. It's not even one of those that actually cause issues, right? You're not giving me against something that's rare. Did you get the idea? Is it against the wild form that we all know or not? It's going to actually protect. So if it is actually can work and protect against the exposure to the natural wild form, that's fine. You don't give me something that protects me against something that we don't even see it, right? You get the idea? So this is another thing. The other thing is, does it stimulate the whole scenario, both T and B, both cell-mediated and antibody-mediated or not? And what do I mean by that? I mean, maybe you give me something that's going to, the macrophage will do it, and it will get to the T helper cells and activate T, and that's it. No, reject it. You have to activate both of them. So I have to follow it all the way. It should have a long term. So you don't give me something and you say, this is going to protect you for a week. A week? You're vaccinating me for a week or a month? Definitely not. You can say, okay, that's going to protect you for a couple of years. That's good enough. Okay? So this is a long term. The doses and boosters should be minimal. Like, I gave it to you today, and then you come back 5 years later, I give you a booster dose. That's fine. But you don't tell me that you come every 3 months or 6 months to get a booster dose. No, that's bad. That's not effective. Come up with something else. Okay? The minimal dose. You don't tell me that I'm going to give you all that injection in order to give effect. No, you have to give me something that a little bit of it is good enough. Okay? It should not be expensive as well, and have a long shelf life. Meaning, you don't give me something and you tell me it has to be used within 3 months. What? You give me something that stays for 5 years or something. We call that the shelf life, right? You give me something, I'm going to buy it from you, or I'm going to approve it, and then you're going to sell it. Whoever is going to buy it should be able to keep it for some time, for a long time. We call it the long shelf time. And you don't make something that you tell me this vaccine will cost you $1,000. What? No, that's not effective. So we have to go through this list. Is that clear? This is a list that you have to follow in order to give an approval, and you say this is an effective vaccine. For the life attenuated vaccine, there is an advantage for that. Like if you ask yourself, if the life attenuated vaccine, this is the actual virus or the actual bacteria, and it is live. Isn't that going to make a stronger response? Yes, this is true. But there are disadvantages to that. So what are the advantages? The microbe is actually viable. So the microbe can actually multiply a little bit, enough to make the infection. So you're making sure that you actually get the infection. If it is killed, yeah, most of the time you get the infection, but I'm not sure if everything will follow through. So it is better to use it, but there are disadvantages. Okay? Being the actual organism, it's going to make a long-lasting protection. It's give you longer time of protection because I'm giving you the actual microbe. The doses and boosters are less because I'm giving you the actual microbe. You eliminated the virulence factor. This is before you give it. What are the disadvantages? The problem is if this is a living organism, even if it's weak, it needs a special storage. You cannot put it on the shelf. Like if this is recombinant, right? You make it from scratch. You make it from chemicals, right? I can put it on the shelf just anywhere, right? But if this is live attenuated, it requires special storage. The other thing is it can be transmitted to other people. Like if I give you that and it activates a little bit, you can infect other people, right? Versus if this is a capsule, a part of the capsule, for example, how can it go to other people? Did you get the idea? The other thing is it can mutate. Remember that, look at this. You eliminated the virulence factor. You know what's the virulence factor? The virulent factor is not the infection, it's about the pathology. Do you get the idea? Virulence factors are the factors that enable you to cause pathology, to cause harm. Do you want to make harm? No. What's the difference between infection and pathology? Isn't that different, two terms? Infection is to stimulate the immune response, that's it. Pathology is to cause harm. So before you give me the live attenuated organism, you need to take off the virulence factors to make sure that it's going to cause infection but not pathology, right? What if it mutated? Like I take this bacteria, I take the virulence factor, and I give it to you, right? It's supposed to just cause infection, right? Not a pathology. What if that bacteria changed and mutated and regained its virulence? It's going to cause disease. This is one of the disadvantages. How about the acellular vaccine or subunits? This is something else. This is something, it shouldn't be here. This is something that have the antigenic determinant of the pathogen, but not the whole cell or virus. We talked about this before. It's not the actual virus, it's just a component. These are the actual licensed vaccines. It's too much to memorize. I'm not asking you to memorize it. Just take a look at it. Okay? It's good to know. Here is live attenuated vaccines. It's something like TB, typhoid. These are examples of live attenuated. Acellular is like anthrax, meningitis, meningococcal and hemococcus. So this is basically you're taking part of the virus itself. Like meningitis, I just take part of the capsule. Okay? Pneumococcal is the same idea. Toxoid, you get the exotoxin and you inactivate it. So what's toxoid? Remember this? Toxoid is inactive exotoxin. So you call it toxoid. Tox means toxin. Oid means similar to, because it's not the actual toxin. It's inactivated by formaldehyde. And the example of that is the DT that a lot of us take it, right? This is toxoid. Whole virus but inactive, hepatitis A, influenza, poliomyelitis, rabies, live attenuated virus also like adenovirus, chickenpox, influenza. So influenza can be any of those, mumps, measles, the MMR. Subunit is like human papilloma virus. Some others are combined, like the DPT. And I think we all hear about this, right? The DPT is a combination. Instead of giving you diphtheria only or tetanus only, I give you DPT, just one vaccine and you're done. Okay? Sometimes I give you hepatitis A and B at the same time. Sometimes you do the DPT and polio hepatitis at the same time. Just go and take that combination. MMR, I think we're familiar with this. MMR? Yes, measles, mumps, rubella. Sometimes you add chickenpox to it. Okay, can you tell me the answer for this question? Acellular. Yes. Herd immunity. Herd means like population. Herd. Okay? A population of something. Herd immunity occurs in a population in which large percentage is immune. What does that mean? Means it's less likely that non-immunized person will encounter the pathogen due to the immunity carried out by the major part of the community. Here is what I mean by the herd immunity. DPT, for example, is a vaccine that most people take, right? So what are the chances that you get diphtheria or tetanus? Minor. Why? Because most of the people are already immune. So we call that herd immunity. So what if did you get the idea? If I didn't take the vaccine, what are the chances that I get sick? Minor. For DPT. Why? Because most of us are already immune, right? So what are the chances that you're sick to give me? You're not sick because you took the vaccine, right? And you're not sick. What are my chances to take it? Very minimal chances because everybody's already immune. We call that the herd immunity. Am I immune? Did I get the vaccine? No. It's just herd immunity. Did you get the concept? So if a lot of people are immunized already, the non-immunized will be kind of immune. Did you get the concept? We call that the herd immunity. Okay? And that's it. The herd immunity is the last part of this chapter.